since we're able to keep the cells alive, for the first time, we can actually observe not just the final outcome, but also the historical process that led to that outcome. And we can start to build models from a very early stage in a process to predict what's going to be the final outcome. And that's actually very exciting, not just from a perspective of shrinking the iteration cycles, but also from a perspective of opening up new kinds of scientific questions that could be asked.
Hi, everybody. Welcome. I am Mark Jacobstein, the co-founder and general partner at Near Horizon, and this is the Near Horizon podcast, where we interview world leaders on AI research, entrepreneurship, and AI ethics about opportunities that are on the near horizon. I am really excited to have my friend Frank Lee with me here today. I've known Frank for a few years. We've been working together on his project, Stately Bio, for some time. Frank's been at the forefront of AI and its intersection specifically with biology for much of his career. After undergraduate and master's work at Stanford, where apparently he had the highest GPA in the School of Engineering. So Frank is one of those. Frank worked everywhere from Calico to Palantir to Google, and most recently at Calico, where he ran all things ML before founding Stately Bio a few years ago. So Frank, welcome. Really excited to have you here. And maybe I'll start with, if you don't mind, describe for us what it is that Stately Bio does.
Yeah, so thanks, Mark, for the introduction. Very excited to be here. Stately Bio is a machine learning company that is actually building cell therapies. And what I mean by that is we have developed some proprietary algorithms to actually inspect images of cells. And then we use that capability to actually improve our ability to use those cells for therapeutic applications. So it's a bit of a hybrid of the tech mindset with the biotech mindset.
And one of the ways I think about this is that in the world of biology, an enormous part of the challenge is simply understanding what's actually happening, right? Cells are tiny. There's trillions of them. They are interacting in extraordinarily complex ways. And I think sometimes, people whose experience with biology ends with like a textbook think that we sort of understand how all these things work. And of course, the reality is we don't. We're trying to figure it out. And we're constantly improving the microscopes, as it were, the technologies for examining what's happening. And so we have things like single cell genomics and ability to pull apart all these very exquisite interactions. But one of the things that's really interesting about Stately is that you're using older school technology in terms of the ways that you are examining the cells in conjunction with the most cutting edge machine learning to understand what's happening with these cells. And so maybe walk us through a little bit, what is brightfield imaging, why you use it, and what the advantages are over, say, more expensive, newer, cutting edge technologies that people are also using as they try to understand biology.
Yeah, great question. So let me start maybe in a different direction, where biology is the study of life, after all. And it's kind of ironic that the current day modern tools that you mentioned, single-cell RNA sequencing, qPCR, all of these other ways of interrogating cell states require us to study cells that are dead, essentially. And that's one of the main challenges, one of the main limitations for actually understanding and unraveling the temporal dimension of biology. What brightfield imaging is very much the earliest form of microscopy that was invented back in the 1600s by Leeuwenhoek. You shine light at a biological specimen, you collect images on the other side, and you can use that to understand the biological specimen's morphology, their live dynamics, and their behavior over time. What's attractive about brightfield imaging is that we've really come a long way in miniaturizing, understanding optics, being able to capture images at very high speeds and very low cost, to be able to actually model all of that biology in a live cell format. That's what is mostly attractive about brightfield imaging, is that it's so able to keep the cells alive without having to perturb them.
I love this notion that we're using technology from Leeuwenhoek in the 1600s. I mean, I appreciate that the lenses are better and that we have improved certain things, but in many ways, it really is. It's sort of the same. It's the same technology. When you were in elementary school and somebody first gave you the simplest, cheapest microscope that exists, where you're shining light through a sample. This is what brightfield imaging is, albeit with better lenses and higher speed photography, et cetera. Is that right?
Exactly. And, there's no lasers involved. There's no fancy optics, really. Obviously, the lenses have gotten improved over the last few centuries. But the fundamental premise of it is really the same as it has been for centuries. Where we take a different spin, though, is coupling that kind of centuries-old approach to imaging with a very modern new approach on the machine learning and analytical side to be able to read out insights that are captured in those images. For many years, a lot of biology has been learned by looking through a microscope. And all of those observations have been, for the most part, qualitative or manually quantified through the use of a lot of labor. And what we can do now, using the advent of modern computer vision techniques, is to automate that quantification process and to reach further into the subtle signals that are present in those images that the human eye can't even readily perceive. And that allows us to actually push that forward.
Well, I think the subtle signals piece is really important. So the automation, of course, matters because it means you can do lots of it and you can do it very fast, and you don't have to hire an army of poor underpaid grad students to do this for you, which is important. More important, I think, is a sort of imputation that, like, one of the things that we have discovered is that machines are better than people with vision at pulling out very, very, very subtle signals. And I mean, I think the first hints we saw of this in biology were probably the Nature paper where we first read out that the machines were better than humans, better than radiologists at understanding what was a malignant skin cancer and what was not, for example. But we've taken that much, much further. And so you're able to do this with living cells. I think it's a really fascinating coupling of obviously cutting edge ML and centuries old technology. I want to go back to something you said earlier, though, that I think is really important to make sure that our listeners understand the importance of this. Most of the modern techniques with sort of multi-omic, super advanced single cell, we're going to look at transcriptomics and proteomics and exquisite ways that we can understand a cell right down to a single cell, require you to kill the cells. And you get a time slice and the cells have sort of just been killed. So you are seeing an accurate, typically time slice of what the cells look like right now, but you can't follow them over time. So you mentioned part of what you're doing is bringing the temporal nature back into sort of understanding biology. So what are the implications of that? What does that allow for that you couldn't do with, say, a multi-omic approach using 10x equipment, single cell work, for example, that now you can do?
Yeah, two main things. One is with regards to the real-time monitoring of live cell cultures. You can essentially have these living cells be imaged and then analyzed in pretty much real time. And that gives you just a different lens into, for instance, if you're trying to use these cells in a manufacturing process, what is actually happening at the cellular level? How do you want to actually take controls and reactions to what is actually happening in the culture to steer things back on course if things are starting to deviate, right? There's a lot of real-time process control aspects and applications there. But another one that I think is actually very exciting is our ability to start to make forecasts into the future. Since we're able to keep the cells alive, for the first time, we can actually observe not just the final outcome, but also the historical process that led to that outcome. And we can start to build models from a very early stage in a process to predict what's going to be the final outcome. And that's actually very exciting, not just from a perspective of shrinking the iteration cycles, but also from a perspective of opening up new kinds of scientific questions that could be asked. And so that's something that we think is really exciting to push forward with this kind of technology.
That's incredibly interesting. What are some of those types of questions and what are sort of the applications of that in the stately bio context?
Right. So for instance, with regards to variability, this is a key challenge of the field in general. And in particular, we've collaborated with partners who, despite being experts in their field for working on specific stem cell differentiation protocols, they still see a lot of variability. And there's a lot of unknown knobs that might have to be tuned just right at the very beginning to get the right result. Now that we can actually build these kinds of forecasting models very early on in the course of a differentiation, we can predict and forecast what's going to be the outcome at the very end of a very long process, oftentimes multiple weeks. And that allows us to actually go and for instance, understand better what are those knobs that we should be turning and what are the right settings for those knobs very early on.
So let's unpack that a little, because this setting and tuning the knobs piece in a stem cell differentiation context turns out to be incredibly important. And I'm ahead of my skis here scientifically, but general understanding is much of regenerative medicine, which is a field where you're doing a lot of your work, requires ultimately the creation of certain types of cells. And a lot of that work happens with stem cells. And the whole point of a stem cell, right, it can become any type of cell, and which cell it becomes and which subtype of cell that it becomes and exactly what characteristics it has is a function of the milieu in which that cell sort of exists. The milieu is essentially sort of the knobs and dials that you're talking about. And if you can change the protocol for the way that these stem cells are sort of exist and then differentiate, you can end up with exactly the cell that you want. And what's an example of the kind of cell that you might want in, say, a clinical setting? Why do we care, other than sort of scientific curiosity? What are some of the clinical applications of this?
Yeah, there's lots of cell types that are, some already in clinical trials. So maybe that's the easiest starting point. There's work being done in pancreatic islet cells being used for type 1 diabetes and eventually maybe type 2 as well. There's differentiations of stem cells into specific kinds of neurons called dopaminergic neurons that are being used for Parkinson's disease. And there's a lot more experimental work being done to try to boost the functional maturation of various cell types for the heart, for the liver, for the kidney, where you can imagine, transplantation alternatives if those cells are viable.
So the downstream clinical impacts of this sort of work range from everything from we no longer need to transplant to save somebody to we can potentially cure diseases that today we really have no cure for. Maybe they're managed diseases, and in some cases, maybe they're not even managed diseases. So really, really exciting work. Let me change gears here for just a second. Your background, your undergraduate and master's work was in computer science and machine learning. Why biology? What was the path?
Yeah, so actually, this is just one of the serendipitous moments. Back when I was in my undergraduate at Stanford, a professor came over from MIT. His name was Drew Endy, and I stumbled into a synthetic biology class that he taught. It was just
not literally stumbled.
Well, I kind of signed up for it because I was curious about it, and I didn't realize actually that it was one of those classes that was a required class for a genetically engineered microbes contest. And so I was the only person who was not on that iGEM team, as it was called, and approached it from kind of a computer science perspective. It was just fascinating to me to see the parallels, though, coming from this outsider's lens between, the world of code that I could engineer and this world of biology that was, being discussed about being able to be engineered. And yet, there's so many parallels and also so many non-parallels where biology is just so much more messy than the abstractions that we've developed in the software side. And so I'd always been really interested in biology. I even considered double majoring in bioengineering as a result of that, but decided that, this was really the area that had the biggest societal impact. We are all biological beings after all. And we can't escape that, at least not until some singularity moment or something like that. And so for the time being, it's going to be an incredibly important endeavor to better understand our biology and to be able to treat the diseases that ill us.
Yeah, that's great. I have a similar journey myself. It's sort of at the intersection of, this is incredibly interesting. The most extraordinary data that we know of sort of in the universe probably is biological data. So if you're into machine learning, what a great thing to work on. And it's intrinsically embedded with meaning. If you discover things with, particularly with human biology, but really biology in general, the implications for human health, what we do with our environment, how we grow things, are all so profound. So you sort of get a triple whammy. And I know a lot of people like yourself who started out in CS and have found their way into sort of bioinformatics or biologically inspired work in ML, and every single one of them, I think, just finds it fascinating and wonderful. I would go back to one other thing we talked about, which I think is often underappreciated in the world of therapeutic development, et cetera, which is cost. So brightfield imaging is cheap. I mean, not cheap, but pretty cheap, and very, very cheap compared to single cell genomics and all the things that are happening with proteomics, transcriptomics, spatial genomics, all of the sort of advanced omics that you hear about. There's lots of very interesting papers. They're very, very, very expensive and difficult. And I think one of the things that leads to it's not just that there's cost to a pharma company that's doing the research, let alone to some academic whose budget is 1/100th of a pharma company's. It's that there's a sparsity of data. Because there just aren't that many cells. If you look at the total number of cells ever that have gone through a single cell multi-omic experience, there's just not that many, not by the standards of machine learning. And so I think that's another component to what your approach that I think is really interesting and important. I don't know if you've seen that play out in some of the commercial work that you guys are actually doing yet.
Yeah, absolutely. We're orders of magnitude lower cost per cell compared to single cell RNA sequencing, for instance, which is a common way to profile cells. Now, obviously, we get different readouts, so it is different, right? What we get are images and machine learning annotations on top of those images compared to the deep molecular readouts that you get from single-cell RNA-seq. But there is a value in being able to capture the heterogeneity of biology. And the only way that you do that is by sampling a very large number of cells, which is something that is eminently doable with these kinds of imaging-based approaches. Right? So
I think one of the things that's most interesting is that in order to do the best work, you actually need both, right? Because part of what's happening is you can have a deep understanding at a snapshot level of individual cells with a technique like multi-omic single cell sequencing or something where you understand exactly what's happening with all of the cells in a particular scenario. And then you overlay that with what are you seeing if you're just taking an inexpensive brightfield image. And part of what happens then is that, and this is the glory of machine learning, is the algorithms are able to impute things. So if the cell kind of sort of looks like this, apparently what that means is the transcriptome is operating in this way, and this is what's happening in the proteome. And no human could ever pull this apart, but the signals are there. They're just incredibly subtle. But with enough cells, we can sort of pull that apart. So you have to have, in order, I feel like, to do the really good training, I don't know if you have to, but it's certainly helpful to have very, very deep data. And then on top of that, you lay across the brightfield data. And we're seeing this sort of imputation, obviously, in lots of different fields, not just biology. They do this with census records or data that's missing in healthcare, in EMRs, what have you. And I think it's a really powerful technique. Do you require deeper data like that, or are you able to do this in sort of a unsupervised way? How does that work?
Yeah, so we can integrate with some of these other data sets. I actually think most of the interest that we've been focused on has been in bypassing those and jumping straight to those assays that are actually proximal to the biology of interest. And what I mean by that is functional assays. So think metabolic assays, things that are extremely expensive and extremely low throughput. If we can actually capture the functional behavior of these cells in a way that is directly meaningful, right? In machine learning parlance, this would be the objective function is closest to what you actually care about there.
Right. So what's an example of the objective function in a biological setting for like a cell therapy or something that you're trying to think about?
Yeah, sure. So for instance, in the context of a liver cell that has not yet been able to become fully mature, which is one of the key bottlenecks of the current state of the art for those protocols, a functional assay could be, for instance, the cell's ability to metabolize specific drugs. And there are quantitative ways to measure that activity and to be able to say, can we actually learn correlations between specific morphologies and other features available from imaging to predicting those kinds of functional readouts. In a way, the way that we're taking this approach, it bypasses the need for some of these other alternative characterizations, only insofar as we're using this as a way to rank hypotheses, to screen spaces more efficiently, and to be able to make improvements on those protocols to actually get to the end outcome that we care about. We would still go back and characterize those cells with, the deep molecular characterization to fill out the picture once we've actually honed in on those specific characterizations that we care about. But being able to use imaging upfront is very, very efficient.
But you might save somebody literally orders of magnitude of sort of number of experiments needed to run, et cetera, in order to get towards the protocol, for example, that's going to push a cell into a state that you want it. So it's going to metabolize a drug, or it's going to have some sort of characteristics that's specifically clinically useful.
That's right.
Very, very cool. So speaking of clinically useful, I understand that you have some very exciting news in regards to Stately, if you'd like to share with our listeners.
Yeah, so we recently raised a fundraising round, and we are displaying some of our work at the ISCT conference. This is the International Society for Cell and Gene Therapy, where some of our technologies have been demonstrated in a couple of different applications. One is a partnership that we have with the New York Blood Center. There's some work that we can talk more about there. And then also internal work on a hepatocyte maturation program, where we've been able to show substantial improvements over the state-of-the-art hepatocyte differentiation protocols, up to 10x better than the state-of-the-art on some of these functional assays that I was just describing. And so we're very excited about the applications of these technologies to being able to break new barriers in the scientific domain, and are very excited to see where this technology holds for the future.
That's fantastic. And of course, it's great to have the capital in order to continue to pursue this. Tell us a little bit more about Stately's business. How many people work at Stately? How did you get Stately going?
Yeah, so we keep a very small group. It's a small but mighty group. Right now it's 4 full-time. My philosophy here has been really to find those individuals who are extremely motivated, extremely capable, of course, and to build a kind of an elite team around that. So the business got started with me as a solo founder. I was trying to find someone, kind of a counterpart to myself on the biology side, given my background on the computational side, and really held out until I found the right fit, and have now been able to grow the team to 2 on the computational side, 2 on the wet lab side, and we've been able to do quite a bit with a small team.
It's one of the interesting things that we're seeing more and more of with particularly sort of ML-oriented startups, that you don't need huge teams. What you need people who understand the computational side deeply, understand what are precisely the right algorithms to be applying. But the tool stack is now so good that you don't need an army of software developers and DevOps people and all the rest of it to sort of keep it all going, right? You just need the folks who understand the stuff algorithmically. And then similarly on the wet lab side, people have to run the experiments, but that's one of the points of Stately, is that you're using relatively simple technologies, relatively inexpensive. So all of this, of course, makes for, I think, a very sort of cost-effective way to pursue deep biological insights, which hopefully is means you can pursue more paths and more drug programs, et cetera, for the same amount of money. It will be interesting, of course, to see how Stately develops. I don't know your thoughts in regards to, we have a platform for discovery, or are we going to build our own drugs, or we're going to partner with other people to make drugs, or how you think about that? Yeah.
Truth be told, I think many companies in the platform space are also trying to figure out those kinds of business models that can be in a sustainable generational sense. The path that we see right here is to use the platform technology to actually make new therapeutic programs. That is something that has inherent value in the field, and also I think is a great demonstration of the capabilities of this platform technology in ways that I think stretch the imagination, otherwise might be hard to conceive. And so by demonstrating that we can use these technologies directly to lead to, for instance, a cell therapy program, that I think is the best way of validating the platform that we could actually take at this point.
Yeah, I've seen many other businesses that are sort of similar to your own, where there's sort of an ML meets biology powered platform where they have sort of come to the same conclusion that you do, which is we can't be just a platform that has no pipeline and has no therapeutic programs, because most of the value capture happens in the creation of the therapeutics, but also to the extent that we want to license this to other biotechs or to larger pharma, et cetera, the way that we can prove that the platform actually has value is by developing a drug. And it doesn't have to be all the way through the phase 3 and FDA approval, et cetera, but it has to be sort of far enough along that you have demonstrated the power of the platform beyond a few posters at at good conferences. And I think it's a conundrum, because we talked earlier about how you can run really lean and have a team of 4, 5, 6 people, which doesn't cost that much money, and still be doing really profoundly important work. Once you move into we are developing therapeutics, there's real cost there. There's sort of no way around. You got to run the animal models, so you got to do the studies. So it doesn't mean you have to take it through a phase 2 or a phase 3, but even to get through IND and into phase 1 is an expensive process. So first of all, kudos on the fundraising and the ability to push forward. That's really exciting. And I'm not surprised you sort of landed in this place where developing therapies, or at least the beginnings of the development of therapies, is an important part of what you're doing.
I would add on to that one thing, which is, we do see this as a platform technology company at the end of the day. Right. And so in addition to the therapeutic work that we're doing, and it would not just be in a single cell type, we have aspirations in future years to actually go and expand way beyond therapeutic applications. Applications in biomanufacturing, for instance, synthetic biology. There's lots of different verticals where the same premise of non-destructive live cell imaging based readouts can be extremely useful. And so that is not lost on us at all. We just think that right now therapeutics is the right path to validating.
Well, that'd be great, given that this all started with you wandering into a synthetic biology class. So that would talk about some of the biomanufacturing applications. What would those look like?
Yeah, so for things like process development or cell line development, there is some intriguing work now, publications that have come out suggesting that, again, even morphological clues that you can get from imaging can be highly predictive of specific cells with high titers or high specific QP for the production of specific biologic molecules. There's applications, obviously, around the process development piece of optimizing the specific media conditions that you might want to be using for growing your cultures. That's more akin to some of the cell therapeutic work that we're doing. And then even more downstream of that, in-process control, as I was saying earlier, in the manufacturing GMP suite, you might want to be able to have these kinds of technologies to provide real-time monitoring and feedback on the status of your manufacturing processes, to alert you into deviations from a baseline that you care about, and or to be able to suggest course corrections to those manufacturing processes.
Very cool. So another question for you. You started your career as a developer, right? Literally writing code, and then a principal engineer and managing larger teams. Now you spent the last few years as a CEO, and obviously it's a different role. Talk about that journey. How did you sort of learn to be a CEO? What have been some of the challenges? Anything unexpected for you along the way with that journey?
Yeah, I guess my first journey here was my first taste of startup life, I should say, was when I co-founded a mobile payments company back when I was in college still. And, me being at Stanford and
I don't think I knew that, but of course you did. You were at Stanford, right?
Yeah, right. It's like one of those things that you just have to do at Stanford and being kind of in Silicon Valley. So for me, that was actually a very key learning experience around the importance of culture and just kind of how to get the early stages going in a way that is setting the company up for success. And then, I did my tour of duty at some of these larger big tech companies, learned, practices from them with regards to, what is good software engineering? What does it actually mean to operate effectively on large teams and coordinate across, large projects so that you can do things that a single individual can't do. And then as I mentioned earlier, transitioned over to the life sciences sector, where I saw there being a lot of room for taking these kinds of approaches that have worked really well on the tech side and applying it to the study of basic as well as translational research. The journey to CEO, I think, was just me getting bitten by the entrepreneurial bug again, honestly, and feeling like there was a big opportunity here that I, in my time at Calico, had seen some glimpses of being really powerful. And kind of taking it to the next logical conclusion would be to further develop these technologies in a direction that I thought was incredibly important for the field, which is this ability to capture the temporal dynamics of biological systems. What that's actually looked like from a personal perspective has been a lot of conversations with fellow CEOs who have walked the path before me, who have graciously served as mentors to me. Lots of kind of research online with regards to what does it mean to do XYZ that I've never thought about before. And I think the main characterization that I would give for how it's been so different now is you just get exposed to so much more about everything about what it means to build a business, right? Everything from IP strategy to how to set up, compensation strategies to how to actually develop technologies and sell that in a way that appeals to the market, to managing investor relations. It's just the whole package, right? And it's just so
it's great for somebody who's infinitely curious.
Yes.
As long as you'd rather be scared than bored, is one way somebody wants to describe.
That's a great way to put it.
Describe being a CEO to me. I do that. It's very interesting to me too because you came of age in a milieu at Stanford in the Valley. Post Y Combinator, right? In a world where there are sort of infinite blogs with good advice and an infrastructure of helpful mentors and people who have sort of walked the path before, it's still hard. The sword of Damocles is still over your head, but at least I think you have some sense of what's coming because of what you could read about it. It's funny. I have this conversation because I'm old. I built my first company in 1994, and sometimes talk to my co-founder Scott about this. That when we encounter entrepreneurs today, like a 19-year-old entrepreneur that we encounter today knows so much more about building a business than we did when we were 28 or 29 and we're 4 or 5 years into running our company. Because there just were, first of all, we happened to be in New York and not in Silicon Valley, and Silicon Alley at that time was not as developed, and there wasn't the milieu, but also there weren't the blogs and the books and all the rest of it. And it doesn't make it easy, but it certainly, I think it's helpful and encouraging. And I'm so glad that, you've taken this path and found it really interesting and exciting.
Huge debt of gratitude to, being able to stand on the shoulders of giants.
Yeah. What's surprised you about the journey?
One thing that surprised me here, probably the most that I continue to maybe grapple with, is there's a lot of opinions out there, oftentimes conflicting. And if you want to find some perspective to validate a particular kind of a stance, I think you'll be able to find it out there. And so it's really your job as a CEO to filter out, what is the right signal from the noise. And it's not very clear all the time what that should be, right? So it's that kind of that taste, if you will, for what is the right path for this company given this stage, given this particular situation, where a generic piece of advice might be generic and not highly adapted to your specific needs. That's something that I think came most clear to me in the most recent few months as I was thinking about this business model question of how to actually build a sustainable generational company here. And there are many opinions on, go one path and many opinions on go the opposite path. And as a CEO, you just have to make those hard calls yourself, informed, of course, but ultimately you have to make those calls.
I think it's very wise. And I will say, I think part of the role of folks who are doing the advising is to understand where they have expertise and where they don't. And hopefully your better mentors and advisors will tell you, you can ask me my opinion on that, but my opinion is not particularly useful here. On this other subject, I have a lot of experience and it's actually quite germane. So pay attention. And I know when I'm going to pound the table is in regards to the advice I'm giving versus, my opinion is no better than anybody else's. It is as a result occasionally a very lonely job though, because you are the one who makes those decisions and there's only so much time and money and what have you for the business to ultimately succeed. So again, kudos on the success with Stately so far. Anything else that'd be interesting for our listeners to hear about regards to the work that you're doing at Stately? I don't know if you have any papers that by the time this comes out in early May, will have dropped that you'd like to talk about, or results that are interesting, or even ones that have come out in, let's say, the last 6 months?
Yeah, I will. So I'll share one thing that we're presenting at the ISCP conference, which is in our partnership with the New York Blood Center. This is something that people did not think was really possible, right? And we're fortunate to have been able to find visionary partners at the New York Blood Center who at least thought it was worth trying. But the ability to actually identify very specific cell populations through imaging. In this case, it's a very different kind of cell, which is technically known as suspension cells. These are just cells floating around in liquid as opposed to growing on a 2D surface. There's even less reason to suspect that you could just take those cells, capture an image of them, and read anything meaningful about them, right? These kind of blood cells. And, we were able to do so with astonishing high degree of concordance with the ground truth measurements.
Over 99%, I think, if I remember from a slide I saw.
There's different ways of quantifying that. The way that we think about it is concordance against flow cytometry, which is the technique that's most often used to measure those cells, and then also against immunofluorescence stains of specific markers of the cell population. Anyway, without getting into too much of the technical details there, the high level takeaway that I had was, that was surprising to us, to the scientists that we were partnered with. And we've continued to be surprised in a pleasant way with regards to how we can actually use these kinds of technologies in just diverse cell types, diverse use cases, beyond what we thought was even necessarily possible. And so if there's one big takeaway that I would like to leave listeners here, it would be, this is really just the beginning of what I think to be kind of a phenomics revolution here, where in the same way that in the 2000s, the genomics revolution really changed the way that we think about how to study biology and how to actually measure biological state. That's really the core of what this company is about, is making a different way of getting that kind of access to biological state in a cheap and efficient manner, using imaging as the primary readout. And we're very excited about that.
You're creating the phenomics revolution. So, and just for our listeners who maybe don't know, you want to just define phenomics for them?
Oh, sure. Yeah. So this is a word that's kind of similar to genomics, being the study of genetics as applied to the entire organism. Here we're talking about the study of phenotypes, or measurements about what that organism is doing functionally, what does it look like, et cetera, to be able to capture a notion of kind of the biological state of that organism. And in this case, when we're talking about cells, what we're looking at is really this temporal dimension that I think is incredibly underlooked and understudied, and also coupled with already a very proven, very rich phenotype of cellular morphology that has already been shown to be highly predictive for many tasks in biology. And so we're just extremely excited about that.
I know it's not a coincidence, but it makes Stately Bio is such a terrific name given the sort of work that you are doing. And it's really exciting. And I think hopefully the downstream impacts here on everything from, biomanufacturing to development of therapeutics is going to be profound. So Frank, thank you so much. I'll ask you one last question here before I let you go, which is any content you've been consuming lately, book, podcasts, blog, that has been really interesting and sort of popped you out of any particular local minima in some interesting way that you think our listeners might enjoy?
Yeah, I've been following the BG2 podcast. This is maybe more tailored around the tech industry and all the buzz around AI and so forth. But I found that the analysis that they've given there, and also some of the people that they've invited as interviewees on that podcast, have just been really insightful for understanding what is the state of the very rapidly changing AI landscape. And that's something that obviously is near and dear to my heart.
Fantastic. Frank, thank you so much for the time today. And it's a wonderful conversation, and really enjoyed our last few years of working together, and look forward to lots of success here for Stately Bio going forward.
Likewise. Thank you so much, Mark.